Recording of neural activity. Recordings can be made chronically after surgery or acutely while anesthetized. Recordings with microelectrodes have fine tips small enough to record the electrical activity of individual's neuron called single unit recording. Recordings with macroelectrodes. The macroelectrodes do not detect the activity of individual neuron but of large number of neurons used when wanting to study a region of brain. In clinical setting, used electrodes attached to scalp produce electroencephalography printout of electric activity. Recordings of neural activity is an understanding of the electrical and chemical signals that neurons use to communicate is essential for understanding brain function. A variety of techniques have been developed to record neural activity, providing insights into the underlying mechanism of behavior, cognition, and emotion and electrophysiological recordings. Electrophysiological recordings measure the electrical activity of the neuron. Intracellular recordings. This technique involves inserting a microelectrode into a neuron to measure its membrane potential and action potentials. It provides the most detailed information about the electrical activity of individual neurons. Extracellular recordings. This technique involves placing an electrode outside of a neuron to measure the extracellular field potential, which is the sum of electrical activity of multiple neurons. Extracellular recordings can be used to study the activity of large population of neurons. Electroencephalography EEG is a technique involves placing electrodes on scalp to measure the electrical activity of the brain. EEG is used to study the brain waves and to diagnose neurological disorders. Magnetoencephalography MEG is a technique measures the magnetic field produced by electrical currents in the brain. MEG is used to study the brain activity with high temporal and spatial resolution. Neuroimaging techniques. Neuroimaging techniques use imaging technologies to visualize brain activity. Functional magnetic resonance imaging fMRI. This technique measures blood flow change in the brain which are correlated with neural activity. fMRI is used to study brain function during the various tasks and behaviors. The positron emission tomography PET is a technique measures the metabolic activity in the brain using radioactive traces. PET is used to study brain function and metabolism. Optogenetics. Optogenetics is a technique that allows researchers to control the activity of specific neurons using light. This technique involves genetically engineering neurons to express light sensitive proteins. By shining light on these proteins, researchers can activate or inhibit the neurons. Optogenetics is a powerful tool for studying the causal relationship between neural activity and behavior. Challenges and limitations. The recording neural activity is a complex task with several challenges. Invasiveness. Some techniques such as intracellular recordings and deep brain stimulation are invasive and can cause damage to the brain. Spatial and temporal resolution. The spatial and temporal resolution of different recording techniques varies. Some techniques such as intracellular recordings provide high spatial and temporal resolution while others such as EEG have lower resolution. Ethics. The use of animals in research raises the ethical concerns. Researchers must adhere to strict guidelines to ensure the human treatment of animals. Noise. Recordings can be contaminated by noise, which can make it difficult to interpret the data. Despite these challenges, the development of new recording techniques and advancements in data analysis method are enabling researchers to make significant progress in understanding the neural activity. Recording the brain's metabolic and synaptic activity. If neural activity of a particular region of the brain increases, the metabolic rate of this region increases. can measure this with radioactive 2-deoxyglucose which enters the cell. After this one done, the experimenter can remove and slice the brain to perform autoradiography locates radioactive substance in a slice of tissue. Another method used involves immediate early genes which around when a cell is activated. Example FOS, a protein producing nucleus of neuron, can be stained for studying. Positron emission tomography used for studying metabolic activity in human brains reveals the location of radioactive traces in living brain. fMRI modification of MRI procedure that gives the measurement of regional metabolisms in the brain.